Hi everyone, I'm Dr. B and I'm here to talk to you today about some of the results of my latest article, Ultraviolet Superradiance from Mega-Networks of Tryptophan in Biological Architectures. Now this work has received a lot of attention lately with over 20,000 article views since the article came out last month and an altmetric score of 175 based on follow-up reports in news articles, blogs, on Facebook and X, formerly known as Twitter, as well as Reddit and a number of videos that have been posted about the contests and some of the implications, even perhaps for consciousness. So I decided, based on the questions I've received, that it would be a great opportunity for me to record a video of my own to talk about the paper and some of its implications. This talk is titled Quantum Effects in Microtubules, Superradiance and the Sensory Motor Response. Again, I'm Dr. Nathan Babcock. I'm a theoretical physicist specializing in quantum biology, and I decided to prepare this video to answer a number of questions and to clarify some things based on a number of comments and questions that I have received about the paper so far. So let's begin. What are microtubules? First of all, microtubules are structures found in cells made of the globular protein tubulin. Microtubules are polymers in the cytoskeleton. The cytoskeleton is the skeleton of the cell made up of actin and intermediate filaments, and the microtubule organizing center found near the center of the cell, near the nuclei of the cell, controls the overall structure of the cytoskeleton in animal cells. Tryptophan is an amino acid. It's a molecule found in most proteins, and it is crucial for the structure of microtubules. We have analyzed a number of networks of tryptophan, the chromophore, fluorescent pigment of the protein, and looking at the structures, we're able to see that the actual tryptophan residues are organized in interesting geometrical networks with significance for their fluorescence properties as well. Now, this work is motivated by previous studies of microtubule excitons. These are electronic excitations that studies have shown can actually form in microtubules. They're believed to be generated by reactive oxygen species. These are molecules of oxygen that have reactive properties when they interact with microtubules. They can create what is known as ultra-weak photon emissions. These are very weak light emissions from the microtubules. Often just only a single photon is emitted during these processes. Now, ROS, that is to say reactive oxygen species induced excitons, are believed to propagate through the microtubules, and that this kind of quantum transfer is understood to be significant in tubulin, where microtubule networks may transfer these ROS-induced excitons, and in fact the photon energy there, that could be relevant for cell signaling. So these kind of implications that were published in the Journal of Photochemistry and Photobiology prompted us to follow up to see if we could learn a little bit more about the scale of the kind of excitons, these optical frequency excitations of the microtubules and what they could be doing, what is their significance there. So we carried out a number of simulations of an effect called superradiance. Superradiance is an effect that can occur in groups of fluorescent pigments. These chromophores can not just produce light on their own, but can interact collectively to produce a kind of collective excitation. And the result is of this is that the radiant properties can then become enhanced, and that they can emit at a faster rate. The image of the microtubule shown in this slide shows you the effect of the delocalization, the spreading out of a single exciton over the entire length of a microtubule. Areas where the exciton is most populated are shown in red, and the lesser populations are shown in blue, but you have an intermediate level of population you can see in green that stretches along the entire length of the microtubule, and this is the kind of coherent excitation that quantum physicists like myself are often interested in. When we look at a single tubulin spiral containing 13 tubulin dimers, this is the basic element of a microtubule that is stacked up over and over again to produce the kind of tubular structure we've seen already on previous slides. What can happen is if now the tryptophan networks inside these spirals and columns of these spirals, the tube itself, are excited, we get this collective emission enhancement. Now we represent this using the capital letter gamma to depict the collective emitter rate and the lowercase gamma to depict the single emitter rate. When we take the ratio of the collective emitter rate to the single emitter rate, we get an impression of the enhancement. And if you look at the single spiral enhancement, when we collect the tryptophan together, all from the tubulin arranged in a single spiral, we see perhaps an order of magnitude enhancement. But if we now line up 99 of these spirals to create a lengthy tube, we can see a real significant superradiance effect approaching 600. So this is more than 2 orders of magnitude increase in the emitter rate based on the collective enhancement versus the rate of a single emitter alone. We continued this study to look at larger and larger structures that are built out of microtubules in the cells. These are kind of structures are sometimes known as the ultrastructure of the cell. And looking at a centriole, this is part of the microtubule organizing center of the cell, we found that collections of microtubules organized in the radial pattern that you can see on this slide, we can actually, in principle at least, achieve enhancements of 3,000 or 4,000 of the collective emitter rate over the single emitter rate. What became interesting in our work was we began to look at the impact of disorder on the system. Of course, physicists like to look at idealized systems, but real life is never quite as ideal as we might like. And so we began to introduce disorder into our simulations of the tryptophan network. And what we found was very promising, because as we increased the size of the microtubule arrangement, the size of the tryptophan network, what we found was that it became more resistant to disorder, that the superradiant effect actually became more robust against disorder as the size of the network increased. Now, we can't necessarily expect that just increasing the size of the network is going to somehow make it more resistant to disorderly effects, but that is what we found in this study. Where we had a significant improvement in the resistance to the disorder term, W, shown on the horizontal axis of this slide, as we increased the size of the centriole, the microtubule arrangement shown here, increasing its length from 64 nanometers all the way up to 256 nanometers. Now, what's most important is that for a disorder parameter of 200 inverse centimeters, shown in the inverse of this slide, we still see a significant potential superradiant enhancement. This disorder parameter is comparable to the amount of disorder you might see at thermal temperatures, a realistic kind of room temperature or physiological temperatures, and the largest centriole we studied still showed at least an order of magnitude improvement in the superradiant effect, demonstrating that at the right time scales, these kind of effects still can be found in potentially living cells. We followed up on these kind of studies, numerical studies, with actual experimental work where we looked at the fluorescence and the absorption spectra of the tryptophan networks, shall we say, in individual tryptophan molecules shown in the orange curves of this slide, as well as in individual tubulin dimers and whole microtubules, which are shown in green and blue respectively. Now, what is something interesting to see here is the Stokes shift. That's the gap between the fluorescence peak and the absorption peak changes as we go from individual tryptophan molecules to tubulin dimers and whole microtubules, indicating that the actual geometric structure of these systems does have a significant effect on the spectral properties of the fluorescence in these systems, again reinforcing the idea that the actual quantum effects that we predict numerically are having a legitimate impact in our actual laboratory experiments. Further experiments that we talk about in the Journal of Physical Chemistry B article that I showed at the beginning of this talk showed enhancements in the quantum yield that would be hard to explain in microtubules without some kind of fluorescence enhancement effect, leading us to conclude that indeed, even at normal laboratory room temperatures, that the actual superradiant effect that we predict from our numerics is providing an enhancement in microtubules in normal ambient conditions. Why is this significant? Well, light and these reactive oxygen species, these reactive forms of the oxygen molecule, have an impact on microtubule structure and their dynamics. Something important is that the Stokes shift, about 1,200 nanometers, or 1 electron volt to a physicist, is comparable to the energy that's already stored in microtubules from their fuel source, GTP to GDP conversion, which is about a fifth of an electron volt. We know that tubulin absorbs and re-emits ultraviolet energy, capturing energy in the near infrared around this frequency range, and that the Stokes shift is significantly reduced in tubulin. So it shows us that the right kind of energies, both in the ground to singlet transition of the reactive oxygen species, in the transition from GTP to GDP, the fuel source of the microtubules, and the actual vibrations themselves in the infrared spectrum of the microtubule are all on the same wavelength, no pun intended. Their frequencies are comparable, showing that these systems, these aspects of the systems, all can interact. Now, why this is important is that when we look at flower petal cells, as you can see here in the electron microscopy images shown, that if you look at the control petal cells, these are Arabidopsis flower petal cells that have been not treated at all. They have kind of a conical shape, as opposed to flower petal cells that are treated with reactive oxygen species. They become more bulbous, shown to the lower right of the two figures here. Now, this is important because we know that microtubules can also be randomized or fragmented, even depolymerized under UV light, and that similar effects happen when they're exposed to reactive oxygen species. What's more is that microtubule growth inhibitors that have been studied in experiments exploring the actual properties of microtubule growth are also tryptophan fluorescence quenchers. They dampen out the tryptophan fluorescence. And though both UV light and reactive oxygen species can produce the same kind of swelling in these plant cells due to their interaction with the microtubule network itself. Microtubules also monitor reactive oxygen species. They're sensors of the cells, indicating a highly intertwined effects between tryptophan fluorescence, absorption, and reactive oxygen species mediated excitations in these cells, which again control both the structure, dynamics, and ultimately the growth of the microtubule networks in these cells. This is highly important because microtubules aren't just structural components, they're active sensor systems in the cell. There's a lot of research that has been shown to indicate that microtubules sense structural tension, sense stress, redox chemistry, and as I mentioned now, light in the cell. Now, they're not just passive sensors, they're active components, and the hydrolysis of GTP, that is guanosine triphosphate, drives phonon vibrations. These are just mechanical vibrations in the microtubules. Now, it was proposed in 2010 that microtubules could undergo a kind of mode locking effect, locking in some modes of these vibrations while locking out others. And this kind of mode locking or phase locking effect is a significant one that's often used in quantum optics laboratories to create ultra-fast laser pulses. So this is a significant kind of effect to look for. And in fact, future studies, well, this is 2021 now, showed that microtubules that exhibit coherent motions and that they can act as optical cavities. And further studies from 2022 have shown that actually there is electron phonon coupling, that is coupling between the electronic, the excited exciton states, and the phonon vibrational states to create what's known as a phanopolaron. So there's a lot of evidence to show that there are the kind of typical quantum optic effects that you would look for in a quantum optics lab in the microtubule systems themselves. So this brings us to the idea of microtubules not just as structural elements and sensors, but as real actuators, sensory motor integrators, or governors, as they used to say in the old days. They are vital to cell structure, cell division, cell motility, and sensing. As I mentioned before, microtubules sense stress in plants. They can sense and respond to cellular tension, and they can actually enact proprioceptive control. That is the cell's sense of its environment. And this kind of cellular perception then proceeds to control cell morphology, similar to the morphological changes in the flower petal cells that I showed on a previous slide. Now, given the fact that microtubules are also good light harvesters, this really ties together a picture of microtubules as sensory motor integrators of mechanical, optical, and chemical information inside and around the cell. This is significant in light of the discovery of superradiance because of the numerous properties of superradiance that have been proposed to be valuable to quantum computing, that is to say quantum information processing applications. So there's a number of unrelated works on superradiance that have shown that superradiance can be used to control and produce individual photons for non-classical light sources, to generate correlative photons, and to produce even so-called superradiant lasers, a new type of laser that is different from the underlying physics of the lasers we have in our laser pointers or in our CD players. Now, even more importantly, that superradiance can now be used to control dissipative dynamics. And this is of crucial importance for quantum information processing applications, where the decoherence of the system needs to be controlled in order to prevent the system from losing the phase coherence, which is so crucial to its operation as a quantum information processing device. Now, the use of the control of dissipative dynamics can also be used to create so-called dark states. In fact, whenever you create a superradiant state, a short-lived, fast-emitting state, you also create a so-called subradiant state or dark state, which is a slow-emitting state, which now can be used for a kind of quantum memory in effect. Now, none of these kind of more quantum information processing effects have been showed conclusively yet in microtubules, but what is important to understand is that the superradiance is a hallmark of all of these effects. And so when we go looking in cells for superradiance, in cells where we already know they have very important sensory motor processing functions, it's very interesting now to consider that the natural consequences of superradiance could be involved in cellular processing as well. Does this mean that there is quantum computing in the brain? For those who are familiar with neurology or neuroscience, you may be aware that microtubules are a very important part of neurons. The highest proportion of microtubules in the body are found in the brain. So some have speculated that the brain could, if it has these quantum effects in microtubules, be a quantum computer. People have asked me, Dr. B, what do you think of this? And I would say, not exactly. I mean, I agree, all eukaryotic cells contain microtubules, and microtubules are most abundant in nerves, and certainly microtubule networks are vastly complex, as you see in the image of this cell. We can see the nucleus in blue and the microtubule network in green, and you can clearly see the kind of center microtubule organizing center that organizes the microtubule network from near the nucleus in the cell. So clearly there's very important processing going on, structural, chemical, sensory, motor processing going on in the cell. Almost certainly some kind of underlying quantum effects driving these kind of properties, but that doesn't necessarily allow us to just jump to the conclusion that the cell is somehow a quantum computer, the way computer scientists think of it, at least. So what does this mean for quantum consciousness research? There's been much said about quantum consciousness research in light of microtubule discoveries. The issue with speculating on the role of these biophysical effects for consciousness research is that consciousness research right now is philosophy, not physics. If you look up the definition of consciousness in the Stanford Encyclopedia of Philosophy, we can see that perhaps no aspect of the mind is more familiar or more puzzling than consciousness and the conscious experience. Now, this is problematic for doing science because currently there is no agreed upon theory of what consciousness actually is, or even necessarily how to study it. So given that consciousness research is currently at the level of philosophical inquiry, it presents challenges for actually doing hard science related to investigations of consciousness. Now, that doesn't rule out scientific consciousness research, but it presents problems for the clarity of that research. When we think of consciousness, there's a number of different definitions that come to mind. There's a so-called kind of ladder of consciousness going from the most primitive forms of consciousness, which is simply presented in terms of agency, the ability for an organism or even a robot to act autonomously. Now, going beyond the simple concept of an agent, we can have something that is called sentient. Sentience describes the ability of an organism to express feelings or even complex emotions. From sentience, we can then consider something like wakefulness, the conscious experience of being awake as opposed to being asleep. And finally, self-awareness, the actual conception of an organism of itself. Now, that's to say nothing of the spiritual, moral, and religious implications of the word consciousness. People often talk about the goal of achieving a higher state of consciousness through meditation or prayer, or even the possibility of achieving some kind of Christ consciousness, cosmic consciousness, which brings us to questions of the ability to contemplate the mysteries of life and the question some have put forward as to whether consciousness is in fact a ubiquitous property of the universe. So regardless of any philosophical, moral, or religious questions about consciousness, as a physicist, I'm just going to focus on the most simple rudimentary one to see if I can get a foothold there, and then develop a theory of consciousness from the most primitive elementary building blocks, which again are the actions that an individual organism can produce in response to its environment. Now, this might be something as simple as an amoeba pursuing, engulfing, and ultimately devouring its prey. So if you take a look at this drawing of an amoeba here and the little bacteria that it is about to absorb, a number of things are going on as the amoeba seeks out and acquires its food, or likewise avoids danger, tries to avoid getting eaten itself. This kind of picture of consciousness as a response to bodily functions is sometimes known as embodied cognition, and of course, much has been said by philosophers about that as well. This brings us to the question of quantum biology and quantum consciousness, and the question of where the two meet, if at all. There have been many conjectures on quantum consciousness, beginning with ideas from John von Neumann and Eugene Wigner, proposing that, in fact, the so-called wave function collapse in quantum mechanics may be caused by consciousness. In contrast, people like Stuart Hameroff and Roger Penrose have proposed things the other way around, that actually wave function collapse through quantum gravity somehow plays a role in generating consciousness. And all this is to say nothing of long-standing debates about the role of consciousness and matter and the interplay between them. The old saying, mind over matter, of course, comes to mind. And this kind of picture of the universe as a mind-matter duality was popularized by Carl Jung and physicist Wolfgang Pauli in their picture of mind-matter duality. Now, the issue here is that these kind of topics range from philosophical theories to pure speculations, and none of these ideas are based on any necessarily empirical evidence, and there's a long way to go before we can actually formulate a proper science to answer any of these questions, if these questions are answerable by science at all. So where does that leave me as a physicist? Well, I've done some research on the current popular models of cognition in psychology and neuroscience. And as a physicist, what appeals to me the most is a study of active inference and what is known as the free energy principle. This is work that goes back to original proposals in thermodynamics from Hermann von Helmholtz, the famous physicist and physician. The free energy principle offers a physical basis for cognition, and it says that self-organizing systems, like living systems, work to reduce a so-called variational free energy. What this means is that a generative model predicts how sensory information is caused, and the causal model is characterized by probability density functions, which of course physicists love to calculate. Inferences drawn from that causal model are used to guide action, whether you are pursuing food as an organism or avoiding danger or seeking shelter. These are all the kinds of actions an organism can take, which need to be done somewhat intelligently in order to survive. Finally, the purpose of this whole framework is that the free energy of the model is minimized with respect to the organism's sensation. So the organism makes predictions about what is going to happen next. It's going to get the food or not. It's going to avoid danger or find shelter or not. And those predictions are then matched against the actual sensations that it receives from its senses. And the predictions are either correct. It predicts it's going to catch the food, and it does, or it doesn't. Ultimately, the goal is minimizing the gap between the predicted outcome and the actual outcome that the organism senses, the organism experiences in some limited or more complex sense. And the free energy principle therefore designates a hypothesis that all physical systems, remember this is physics, so in universally all physical systems that perceive and respond can be constructed in this fundamental way of trying to minimize the free energy associated with the gap between what is actually happening and what the organism can predict is about to happen next. And an excellent article has been written on this by Friston in Nature Reviews Neuroscience, which I can recommend for those who want to look into this further. So finally, what does this have to do with microtubules? We've looked at microtubules, their importance in living cells, and the possible quantum effects that they can harbor, including the influence of superradiance. Now, microtubules are found in all complex life forms where they regulate a wide variety of sensory motor cell functions. As sensory motor integrators, they have many unique quantum mechanical properties ranging from the ability to transport excitons, at least short distances, and their ability to exhibit superradiance effects under the right time, temperature, and length scale conditions. Finally, as I've hinted at, microtubules can enable so-called hybrid quantum effects. The existence of excitons, the existence of vibrations or optical excitations do not happen in isolation in microtubules, but rather there is a hybridization that occurs, a sort of unifying of the coordinates that are used to describe the electronic states, the vibrational states, the polarization states or polaronic states, and ultimately the light matter interactions known as polariton states. So the fact that microtubules can actually unify these various physical properties in their structures suggests an ideal situation where the kind of unification of multiple sensory inputs into a single conscious experience, or at least a unified model of the environment, can occur. Finally, microtubules are known to drive unstable thermodynamics through the hydrolysis of GTP, that guanosine triphosphate, which is hydrolyzed into guanosine diphosphate. Now, what is most fascinating about this is that this sort of microtubule fuel, GTP, drives a process known as dynamical instability, where the microtubule network is continually in a state of approaching equilibrium, but with the addition of fuel remains in this thermodynamically unstable state perpetually. Now, this is exactly the kind of thing we would want for a sensory motor system where it is constantly attempting to, shall we say, reduce the free energy, minimize the free energy, but never actually getting to a fixed minimum because, of course, new information is constantly coming in. So microtubules exhibit the kind of thermodynamic instability that we would look for in a system that is constantly approaching equilibrium, but never getting there because, of course, the free energy that is being minimized is constantly being updated as new sensory information comes in. This creates an ideal picture of a microtubule network as having all the properties that we would need to integrate sensory motor features in a unified kind of mechanical or physical model of the outstanding properties, the environment of the cell, in order to enact a concerted, coherent response to the environmental implications of the cell's environment, the cell's habitat, if you will. So I hope this answers some of your questions about consciousness, about cellular cognition, and what microtubule excitations could mean, what the superradiant effects that we've identified in microtubules, what kind of implications these could have for cellular sensing, processing, and decision making in living things. I'd like to acknowledge my colleagues and collaborators who helped me and honestly really did an incredible amount of great work in the production of this ultraviolet superradiance from Mega-Networks of Tryptophan in Biological Architectures that was published in the Journal of Physical Chemistry B just last month. So thanks very much to them, to all my colleagues at the Quantum Biology Laboratory in Howard University and abroad in Mexico, Italy, and Switzerland, and of course to my colleagues with the Guy Foundation, the organization that sponsored this work. None of it would have been possible without them. So hats off to them. In conclusion, I hope this has answered some of your questions and hopefully proposed a lot more questions to come. I know we have plenty of research to do on this field, and it's very exciting to be able to talk to you about it today. Have a good day.